So I'm Kevin Bolding. I'm a faculty member at Monell and I run a neuroscience lab. We study how the brain represents odors, how those odor representations acquire meaning, and then how those representations are formed through learning over time. And we combine behavior with large-scale neural population recordings and analysis to try to understand how those neural representations actually guide adaptive behavior. I came to study the olfactory system because it's a tractable system in which to study a hard problem, which is how the brain stores and uses information. Animals can learn about odors very quickly, and it's possible to record and observe neural population activity during that learning process. Over time, the type of work has become more and more focused on memory, how odor memory representations are formed, and how they're retrieved and used by the animal. My interest in neuroscience started and continues to be in memory, how memories are formed and how they're retrieved and how they're instantiated in neural circuits. So if you start from that background, your initial interest often lands in the hippocampus or the amygdala, and that's where my previous work was. I studied neural responses in the hippocampus in particular. But many of the same rules and ideas apply when you're thinking about storing information distributed across the brain. And there are particular parallels with olfactory memory that drew me into thinking about the olfactory system in particular. The fundamental reason I'm interested in odor memory is because it's a model for memory overall. Memory is obviously important. We use our memories to make our way through the world. They also make up an important part of our identity. If you want to know about odor memory in particular, of course you need odor memory to decide what to eat, to run away from a fire, all these basic things that you need to survive and be fit in the world. I care most about understanding how information is embedded in brain circuits as a model for understanding learning and memory. In our case, we can watch large populations of neurons acquire information, and the goal is to move beyond learning about how individual responses change, but to link that to how that information is used when an animal is behaving. I tell people that Monell is an independent research institute with a focus specifically on chemical senses. And when you first think about chemical senses, you may initially think about olfaction because that's, you know that you breathe in molecules that make you smell something. But then as you move beyond that, there's taste. And more recently, Monell has expanded significantly in the area of interoception, sort of internal sensing of chemicals, how your gut detects nutrients and things like that. So it's a natural home for the type of work that I do in olfaction. It's the place where I started my first lab, where I'm running a lab for the first time. And there's a collaborative environment here that started to shape the type of scientific questions that I'm interested in, the sort of research I'm pursuing. Your chemical senses affect every aspect of your daily life. At the more fundamental level, the types of things we're studying, how neural circuits process information and store information, how neural circuits work and allow animals and people to behave and do things that are useful is just a fundamental thing that you need to know about. And we want to know about it partly because it's fundamental knowledge. And if you need a reason beyond that, the reason is because this is the type of knowledge that forms the foundation to give you hope for any treatments that are really brain focused. So, we know that, for instance, smell is impacted by aging. All sorts of different functions are affected by aging and neurodegenerative disease. We need to know how the system works if we want to repair it. Everything that you value from science and technology came from fundamental research. And the people who developed that technology did not know they were developing a cure for a disease or a new technology when they developed. They were studying the basic phenomenon, trying to understand how nature and the world works. There's new gene therapy coming online. There's people getting genetic diseases fixed. The tools to develop that were built by people wanting to know how bacteria trade their DNA back and forth, wanting to know how worms share genes between each other. Stuff that to you might sound like it's a waste of time really actually has led, with time and patience and deep investment, to the things that you value now. And the goal is to continue that. The beneficiaries are scientists and clinicians, people who need to know how neural circuits work, how they process information, etc. We're doing the work of expanding that knowledge base. Perhaps far in the future, the people who are intended to benefit are people suffering from issues with the way their neural circuits are working. So once you know how the thing is working, you have the opportunity to make changes to it. To give the example of aging and smell loss, if we can understand how neural circuits are affected by aging, then perhaps we have the opportunity to affect the degree of smell loss or the recovery from it. The underlying problems and questions driving our work are fundamental to understanding how brains work and are therefore fundamental to understanding, health and wellness. Some of my core questions are really about memory. We obviously all deeply value our memories, and we feel that we obviously see the tragedy when your memories start to become impaired. The broader work at Monell is focused on chemical senses, taste, smell, and nutrition. These are really at the center of health over the lifespan, and we would like to have a deep understanding of these processes so that we can impact health outcomes over the long term. When you invest in Monell, you invest in the ability of people to do this work, to uncover fundamental sort of rules and mechanisms and aspects of how the chemical senses work. And when we know those things, we have the opportunity to make advances towards improving health outcomes. My career at Monell was interesting because I knew, quite a bit before, at least many months before, that I would be here. But I sort of immediately got involved with the team and with colleagues here so I could get sort of a running start. So, I mentioned before about sort of people being proactive, helping you with your ideas and helping you set up your first concepts and things like that. I immediately had a team with, 6 other Monell faculty looking at my research proposals, etc. So I felt like I kind of hit the ground running on that front. It was exciting to actually get the space set up. I remember the facilities team really being very helpful, and they redid all the space and made it all modernized and just got us set up so we could get started on experiments pretty quickly. One of the areas of Monell that helped me the most immediately as a new PI was the research administration or grants administration team, because they really proactively guided me through initial funding applications. And not only that, but Monell had an infrastructure for making sure that other faculty are involved, attending to the types of proposals you're making suggestions, critiquing, giving you the feedback that you need to succeed in the funding environment. I get the sense, though, that specifically the idea that there's an organized system for helping with grant proposal success is something really unique to Monell. I've talked to many other, people about my generation in science, and I think they're a little envious sometimes that we have this sort of proactive, guided path to start the, when you launch your new lab. When you start a lab, when I started my lab, I think everyone has the experience that you come in with a very specific set of what's going to be your research focus. And what has happened for me over my time at Monell is that I've gotten exposed to the interests of the colleagues here. I was initially really focused on synaptic plasticity and memory and information embedding in neural circuits. My interactions here have led me to think a lot more about olfactory perceptual space, how the actual chemical structure of the molecule matters. Because in my previous work, it was just another odor and another odor. I didn't even think about what odor it smelled like. It was just a different type of stimulus for the experiment. In olfaction, we want to know what the olfactory system is doing that's useful. And sometimes it's a little difficult to nail that down when you're just asking animals to tell one odor from another odor, or asking neurons to tell one odor from another. But one of the things that every organism uses olfaction for is to decide what to eat and when to eat, and is this safe to eat, etc. The correspondences between sort of my basic interests in what are the representations in odor circuits, how does one odor relate to another, etc. Those have started to be really framed in the context of, how does this relate to food? How does this relate to nutrition? I'm getting influenced by my environment, basically, in ways that give me new questions that aren't necessarily immediate follow-ups from my previous work. If I remain in my insular olfactory neuroscience field, we're just going to continue to refine and refine and refine on this sort of main idea in olfactory neuroscience, which, a lot of it is what really is exactly what the brain says when it means this odor versus this odor. And, how's that actually happen? But when you get the multidisciplinary environment, you get pulled off of that really tiny, tight focus. And I think that's helpful, not just because it opens up novel ideas, but it prevents you from doing redundant effort with the rest of the field. You get to explore a new niche and a new sort of frontier. Yeah, I think it helps define a different, distinctive brand or area for my lab that I wouldn't do if I was really just focused on the next thing after my postdoctoral work. This week, I'm spending a lot of time thinking about one of our core research projects, which is trying to understand what really changes the very first few times you smell a new thing that you've never smelled before. How does a new odor representation emerge in the brain? We've got really high quality data. We've got lots of neurons recorded at the same time in a way that nobody else has been able to do. We can really witness this new memory being formed. And I'm starting to make headway with the analysis to really see what are the changes that occur in the circuit. I think that's an exciting direction right now for me. And we'll be able to link it back through to the animal's behavior to understand, how is it that this new representation actually influences what the animal does with the odor. I think the main thing that we need to do as a field in olfaction is to really understand what natural odors are like. We made incredible progress understanding what the brain does with odors that we can easily manage in the lab. We have a very good idea about that. But the thing I'm excited about now is moving on to the truly complex stimuli that occur in the real world. That fits a lot of other sort of categories of interesting research. So for example, odors of other animals or other people, whether those are your friends or your predators, etc. Other natural odors are foods. Obviously, foods in the wild are mixed with everything else too, and they're hidden behind things. So there's a spatial element to the search for food. How are they found? Where are they found? How are they found? How much of it there is in the world? These are things that we need to really understand. And one other major dimension of this, which I think is probably the one that we're going to make headway on the fastest, is the actual complexity of the stimuli. It's not one molecule at a time. A stimulus is many molecules together. And we're starting to get a grip on what it means to mix together different molecules to make something that's really a unique, complex mixture, like the things that you actually encounter in the world. To really understand how to mix things together and affect perception and the neural responses, I think is going to be the big frontier.